Okay, I think we have quite a few people joining now. So hello everyone to the Helios Invited Speaker Series. Helios, as most of you know, is a 4-year funded project by the Cost Action of the European Union, and it brings together thought leaders and uprising stars in the field of hemoglobinopathies, and together we aim to increase the knowledge and the awareness about hemoglobinopathies to give different stakeholders the tools they need to improve their patient care. And Helios provides funding for networking, training, mentorship, and conference attendance opportunities. And at the moment, we have about 200 members, mostly with a focus on Europe, but we generally welcome everyone from all over the world who has an interest in hemoglobinopathies. The Helios project is generally divided into research coordination and capacity building activities. Working groups 1 and 3 identify needs and then try to harmonize procedures and protocols. Working groups 1 to 3 then also promote multi-center data-driven research, whereas working group 2 then monitors the implementation and reviews new treatments. Working group 5 is then aimed to disseminate the knowledge created. And from a capacity building point of view, the working groups 4 then promote young researchers, women, and upcoming stars in general. Working groups 1 promote the use of new technologies, both for diagnosis and for research. And working group 4 develops new European training programs for the different areas that Helios covers. And working groups 4 and 5 promote cross-sectoral dialogue. In general, the different working groups are either aimed at the knowledge creation, so that's working groups 1, 2, and 3, which then is transferred by working groups 4 and 5. The Helios leadership team is set up by many different experts well known in the field of hemoglobinopathies, such as Dr. Petrus Kontouris, Dr. Raffaella Colombatti, Dr. Babenusa, Dr. Andreas Lenzoy, and many other experts in the field. And again, the focus is mostly on Europe, but we generally welcome anyone with an interest in hemoglobinopathies from all over the world. The invited speaker series, they run on a monthly basis. We already have our 2 next speakers confirmed on the 13th of November. That will be with Dr. Sophie Landskron, who will talk about the cost and impact of sickle cell disease in adult patients. You have to register for this webinar separately from the ones today. And the other webinar in December then is together with Dr. Kristen Howell, who will speak about the difficulties in the transfer from the pediatric patients to the adult care for sickle cell patients. And again, if you have an interest in this webinar, you have to register for each webinar separately. Today, we kick off the speaker series with our first speaker, Dr. Oyesola Ojewunmi. Dr. Oyesola Ojewunmi had a PhD from the Lagos University in Nigeria in chemistry. He then worked for King's College London and the London School of Hygiene in London, and now works for Queen Mary University in London. During his time at King's College London, he led the groundbreaking research on the genome-wide association study focused on fetal hemoglobin in Nigerian patients. And this also then led to the publication that we will talk about today with the name of Genetic Dissection of Fetal Hemoglobin Persistence in Sickle Cell Disease in Nigeria. Okay, so this was just a brief overview. And now, Sola, over to you. Thank you.
Okay, thank you, Stephanie, for your kind introduction. I will share my screen right away. Can you see my screen, please?
Yes, you can go. Thank you.
Oh, okay. Yeah, thank you very much. I appreciate you for your kind introduction. I also thank you for inviting me to give this talk. I will be talking about the projects which we did at King's College London with Dr. Stefan Mentel as the PI of the project, and with the collaboration we had with PIs from Nigeria from 3 study site, and also how we worked with PIs from Tanzania and from the US as part of this study. So I'm going to show the data set in this talk this afternoon from my side. So the title is Genetic Dissection of Fetal Hemoglobin Persistence in Sickle Cell Disease. By way of introduction, I would like to define what sickle cell disease is and then go straight away into the main presentation. So sickle cell disease, as we know, it's an inherited blood disorder, which is caused by point mutation in the beta globin gene. And what we notice is that under low oxygen tension, the HbS becomes polymerized. And when it becomes polymerized, it's in a deformed shape, which changes the usual biconcave disc shape of red blood cell into something that is very rigid. And this is what goes on to occlude the blood vessels and leads to several complications we see in sickle cell disease. Some of the complications we see acute chronic complications such as vaso-occlusive pain. We see stroke in patients with sickle cell disease. We see kidneys being affected. We see leg ulceration in patients with sickle cell disease. Generally, for fetal hemoglobin is the predominant hemoglobin that you see in fetus. And shortly before birth, this fetal hemoglobin declines. And from about 3 months to 6 months, the fetal hemoglobin switches completely from the fetal to adult hemoglobin. And that's what we see by 6 months after birth. This hemoglobin switching is genetically controlled. However, in some individuals, the fetal hemoglobin level is still retained, even when even after the 6 months that you would expect that they should have adult hemoglobin. Okay, so they have adult hemoglobin, and then you still see a higher level of fetal hemoglobin in some individuals, and particularly in patients with sickle cell disease. And this has been shown that individuals with sickle cell disease that have fetal hemoglobin level greater than 8.6, they tend to have improved survival compared to their counterparts with fetal hemoglobin that is less than 8.6. I would like to remind us that sickle cell disease is prevalent in the sub-Saharan Africa, particularly in Nigeria, where you see the largest burden of people living with sickle cell disease. And also you will see it's prevalent in DRC Congo, in Uganda, in Tanzania. And of course, we also have large number of people being affected in some parts of India. So coming back to fetal hemoglobin, the first GWAS of fetal hemoglobin was published in 2007, and this was led by Professor Sulitain, who is currently working in NIH in the US now, but then this was carried out at King's College London, and the first author of the paper was Dr. Stefan Menzel, who I happened to work with on this project, who was the lead, was the PI for this project. So in that 2007, when they carried out this first GWAS of fetal hemoglobin, they did it in twins UK data set, and they mapped it to 3 regions, which are BCL11A locus, the HBS1L polymorphism region on chromosome 6, and XMN1 in HBG2 on chromosome 11. Shortly after that, which was in 2008, these papers came out, and they were showing here that the BCL11A is actually the same thing. The BCL11A, there's an association with, it has an association with fetal hemoglobin in patients with sickle cell disease and beta thalassemia. And then they also showed how it's helpful in mediating painful crisis in sickle cell disease. So what they mapped in sickle cell disease patients were the same thing that were mapped previously in the UK twins data set, which was published in 2007. So from then, different studies have been carried out looking at different populations, investigating the association between different genetic variants that may be be contributing to the fetal hemoglobin expression in patients with sickle cell disease. But like we know, in GWAS, we have several limitations. But one of the limitations is the limitation of the missing heritability. And what this means is that we still have a number of genetic variants that are contributing to a particular trait or a particular disease, especially in complex diseases, which we have not been able to know what exactly what are these genetic variants, but we know that these genetic variants are contributing to this trait or disease. So as you would see in complex disease or complex traits, and in most GWAS studies, you will notice that what you are having from the genome, the heritability explained by the genome-wide significance, and the heritability explained by the entire genome, the entire genome in this, the SNPs in the genome, is still far less than what is the heritability that was captured in the twins data set. So coming to sickle cell disease, we have seen in many studies that heritability that could be explained by genome-wide significance is just about 30%. Heritability, SNP heritability could only be explained by less than 50%. But in the twin study, 89% has been estimated as the heritability for fetal hemoglobin. So what that means is up to about half of this heritability that was captured in twins data set has not, is not known yet. And that's what we are looking at in terms of missing heritability. So while we were doing this study, our focus was in three phases. Number one is that can we find new loci that could give additional insight to HbF regulation in patients with sickle cell disease? Number two, is there a possibility to predict HbF using polygenic curve that we will get from the SNPs that will be implicated from this study? And then number three is, could we, with these two aforementioned goals, could we provide pathways that could be useful in targeting HbF expression in SCD, particularly in the development of small molecules that can be used as therapy in sickle cell disease? And this, in my opinion, will be having a far more reaching effect on patients with sickle cell disease, especially in Africa, where the disease is a major public health. So our hypothesis was that Nigeria, just like many other African countries, has exceptional ethnic diversity, which could enable discovery of new loci, and then could also support in fine mapping different loci, which could be potentially functional. Okay, so that's why we felt it would be important to look at the Nigerian patients with sickle cell disease. So our aim was to carry out genome-wide association study of fetal hemoglobin in patients with sickle cell disease in Nigeria, to search for new loci and to perform fine mapping in order to identify functional variants in persons with sickle cell disease, which we hope will provide further insight into HbF regulation and heritability. So we recruited patients from 3 geopolitical zones in Nigeria, one from Lagos in the southwest part of Nigeria, one from the north central, which is Abuja, and another from the from Zaria, which is in Kaduna, the northwestern part of the country. The study in Lagos was, the recruitment in Lagos was led by Professor Titilope Adeyemo, and then the one from Abuja was led by Professor Inodu, and then the one from Zaria was led by Professor Afsat Ahmad. And we are very grateful for the collaboration we have with them to have access to these patients that we used in the study. So this is the study design in terms of the patient recruitment, how many number of people we genotyped, and the pipeline that we use for our analysis. This is the principal component analysis, which is telling us the how the ancestry of the patients that we are using this data set, how it maps within the global population. So This is the African population in blue, and then this purple color that you are seeing here represents our data set, which is from Nigeria, and then maps nicely within the African data set as expected. But we wanted to look further to see the diversity of our of the patients we have in this study within the African population. So we have, for instance, Ishan and Yoruba Ibadan in the background here. This one, we have data set from Sierra Leone, data set from Gambia, data set from I think this is Kenya. Okay, so we have this data set from 1000 Genome Project, and then we were mapping our data sets within this sub-African populations. And then what we see here, nice, is to the blue color and the black color represents data set from Nigeria. So we have people who self-identified as from Yoruba or Igbo ethnicity, or they are from Edo in Nigeria. And then those parts are within the southern part of Nigeria. And then we have people who self-identify as Hausas or Fulani, which are from the northern part of Nigeria. And then we could see that nicely, that the ones in blue, which are basically those ones who are Yoruba, Igbo within the southern part of Nigeria, cluster differently compared to the one from the Hausa, those who self-identify as Hausa and Fulani from the data set. And this is showing, is illustrating the diversity of the patient's population, even within Nigeria. So what I did first when I saw this was to look at the relationship between the CRISPR component and the fetal hemoglobin to see whether the architecture of the patients that we are using, genetic architecture of the patients we are using in terms of their ancestry, could affect the fetal hemoglobin. So when I checked that, I saw that I didn't affect it. But with this, we could see that there's that diversity even among the population in Nigeria. So these Manhattan plots, which we conducted the genetic association, where we conducted association between our genetic variants and the fetal hemoglobin. So we found BCL11A and HBS1L MIP, which have also been identified in many previous studies, including the study that was carried out and published in 2015 from Tanzania. But we couldn't find another popular locus on chromosome 11, which is XMN1 HBG2. And this is because the SNP is less frequent in the Nigerian population. However, we found 3 previously unidentified loci from this data set. And when we looked at the quick view plots, we wanted to see how the SNPs that showed association looks compared to the null hypothesis. And then we could see the SNPs in this region, and then the lambda, which is showing the genomic inflation control factor of 1.004, showing that the analysis which we carried out properly control for the population structure, and then there is no effect of population structure that could affect the analysis that has been done. So this is a list of all the genome-wide and suggestive loci that we found across the genome, and we try to do replication of the new loci that we found on chromosome 9, chromosome 15, and on chromosome 18. We couldn't find replication for the ones on chromosome 9 and 15, but for the one on chromosome 18, PISO2, we found replication in a subset data set that we collected from the, that we got from the African American cohort from St. Jude Children's Hospital in the US. So only PISO2 was replicated. We further went ahead to do conditional analysis on chromosome 2, and we found another secondary signal apart from the lead SNP, which we found on BCL11A, which now means we have 2 independent signals on chromosome 2 for BCL11A. We did the same thing for chromosome 6. This is the lead SNP we found on chromosome 6, and this is the first time we are finding this as the lead SNP. We didn't find it even in the twins data set, because in the European population, this SNP is rare. And that's what we see generally on HBS1L MIP on chromosome 6. The frequency varies significantly when we compare that to the European population. So we also found two independent signals on chromosome 6, one for RS610-28892 and the other for RS9399137. And these SNPs we found based on the conditional analysis form part of the haplotype analysis, which I will show shortly in my few slides to come. So this is a regionalization plot where we're looking at zooming in into chromosome 2, where we have the BCL11A locus, and these are all the SNPs that are showing the association I showed in Manhattan plot. So as you could see here, there are 3 sites here, plus 55, plus 58, and plus 62. These are DNAs and persensitive sites, okay? And the SNPs we found from the lead SNP to this other SNP, which shows a credible SNP with the lead SNP, and RS1427-407, they overlap nicely with the erythroid enhancer element, showing that these SNPs we have found in this region are likely have the presence, or carry the the high tendency to be functional. Then we did the same thing for HBS1L MIP on chromosome 6, and we saw that the lead SNP and its independent signal RS9399137, they are sitting also overlapping with the DNase 1 hypersensitive site at -84 site, which also shows that they are likely, to be functional SNPs. Now, because of the haplotype analysis that we wanted to do, we needed to do a proper fine mapping. For instance, this lead SNP and RS76173, they are in ILD, okay, with a correlation coefficient of 0.91. So the question is, which has the stronger effect in terms of fetal hemoglobin, in terms of boosting fetal hemoglobin between these two? Okay, because they're in ILD, it's possible it could be RS76173. And in the population data set that we have used, the correlation is very high, 0.91. So we looked at the 1000 genome data set, and we saw that in the European population, the correlation coefficient between these two SNPs is 0.61. It's not as strong as what we will see in the African population. So what we did because of that was to look at the UK twins data set, 5,060 of them. We did GWAS for those 5,000 individuals. And then from that, we could see that RS76648 and RS76173 that we are trying to find map, RS76173 has stronger effect size compared to the RS76648. So for our conditional, for our haplotype association analysis, then we include 76173 and we drop the lead SNP because this may have stronger effect compared to RS67648. The other thing which we did was to look at RS1427-407, still on BCL11A. This SNP has been found by Dr. Bauer et al. In 2014 that it is functional. So we found it as a secondary signal to our lead SNP on chromosome 2. So the other thing we now did was to do conditional analysis to see what other signal can we have when we condition on RS1427-407. And what we found was RS7565301 as an independent signal. So for our haplotype analysis, we now included this SNP, RS1427-407, RS7565301, and the RS760 that we found through the that we find mapped using the Twins UK dataset. And I will show that shortly. So one thing I want to mention here is that in the Tanzania dataset, which was published in 2015, looking at GWAS of fetal hemoglobin, the secondary signal they found for RS 1427-407, which was their own lead SNP for that BCL11A locus, was RS 6545-816, which is different to what we found from this study as the secondary signal to 407. I just wanted to mention that. Now for the haplotype analysis for BCL11A locus. So we focused on these 3 SNPs, and these 3 SNPs, like I mentioned before, they are within the erythroid enhancer for BCL11A. So for plus 55, we have this SNP. We have for plus 58, we have this SNP RS75301, and then for 407 at plus 62 sites. And this forms the basis of this the haplotype that we constructed. And this is a picture of the linkage disequilibrium which we generated from HaploView. So the 3 SNPs we have here is now tagged by 1, the T allele for 407, and the A allele for 301. And then what we found out is that the effect is higher, is significant in boosting fetal hemoglobin level when we have the TGG and when we have GAG, which is tagged by T allele for 407 and tagged by A allele for RS7565301. So when we looked at that relative to the fetal hemoglobin level, we saw that individuals with TGG haplotype will definitely have higher level of fetal hemoglobin, as well as the individuals with GAG. And these are the 2 major drivers for fetal hemoglobin levels in BCL11A based on this data that we did the haplotype assertion for. The same thing we did for dissection of the HBS1 MYB haplotype assertion analysis. And you could see from here that at the MYB enhancer element, we have 2 major loci within this region. We have HMIP2A, which is HBS1 MYB, 2A locus, and then we have another locus, HMIP2B, HMIP2B. And from previous study, we have seen that this region 2B and 2A, they are both important in driving the fetal hemoglobin level on chromosome 6. However, in this study, what we found is that our lead SNP and RS9399137, there are 2 independent signals that we found based on the conditional analysis. And from this, we constructed the haplotype, the haplotype for BCL11A, and the haplotype is tagged by C in this for this RS939137, and then another one haplotype is tagged to C again on RS610-28892. So individuals with GC are expected to have high level of fetal hemoglobin alongside individuals with CT. And that's what we saw when we looked at the distribution of fetal hemoglobin based on the haplotypes. Those with GC and CT have high level of fetal hemoglobin, and those without the C for both SNPs have very low level of fetal hemoglobin. So what we saw here is quite different from what we expected, because we were expecting something that is similar to what was found in the Tanzania data set. In the study they published in 2015, the Tanzania data set shows that yes, there are 2 loci within the HBS1 MYB, which is 2A and 2B, like I mentioned previously. And then the SNP here is being tagged by RS 666-50371. I'm not sure you can see that. And then for HMIP2B is being tagged by RS 9494-145. However, in what I showed, you will see that all the SNPs here, which are representing the HMIP2B, they are tagged by this RS610-28892. And you could see that clearly from this LD relationship. If you look at RS610-28892 compared to these SNPs that are on HMIP 2B region, these SNP are in ILD with them. So meaning that this region is not standing on its own. It is actually mapped, tagged by this SNP. So what that means is that the genetic drivers for fetal hemoglobin on chromosome 6 is tagged only to HMIP2A and not HMIP2A and 2B as we have previously seen. So the next thing we did was to look at those haplotypes if they are replicated in the UKSCD data set, which contains 805 patients. And then we saw exact effect of those haplotypes in increasing fetal hemoglobin level in the UKSCD data set. We saw the same thing on HBS1L MIP, and then which shows that these haplotypes may actually be functional. The other thing which we did was to integrate our data set with the public data set from ChIP-seq GATA1 data set, UDEP2 and K562. These are data sets which have been used previously, which have been generated previously, and they are in the public domain. And when we integrated our data set with GATA1, UDEP2, and K562, we saw that the RS42747 is present in the GATA1 data set, and this is what we will have. This is what we expected, because this SNP has already been shown to be functional as a SNP that is involved in the GATA1 transcription factor site for BCL11A. The same thing we did for HBS1L MIP. When we integrated the data set with GATA1, UDEP2, and K562, and then again we saw RS6102889 as which is our lead SNP. We saw that coming up in the GATA1 data set, which shows that that region may actually be functional. So the next thing we did was to carry out a polygenic score based on some representative SNPs we have seen on chromosome 2 for BCL11A and then chromosome 6 for HBS1L MIP. And then for we had 8 representative SNPs, but after coming up with, after doing the modeling, we only have 5 SNPs coming out, which are these SNPs. And then these SNPs account for 24% of variability of fetal hemoglobin. And when we did the genome-wide SNP heritability, we could only account for 44% based on the data set from Nigeria. So the summary of our findings: we confirmed known assertions for fetal hemoglobin in BCL11A and HBS1 loci, and we found the lead SNP for BCL11A to be this SNP, and RS6102892 for the one on chromosome 6. We detected previously unidentified signals at chromosome 9, chromosome 15, and then PISO2 on chromosome 18. And of course, like I showed the haplotypes that are influencing the level of fetal hemoglobin based on BCL11A and HBS1L MIP, which is significant for this study coming, for this data set coming from Nigeria. We also showed that there is a distinct departure when we looked at the HBS1L MIP in terms of the drivers for fetal hemoglobin levels in patients with sickle cell disease when we compare our data set with the Tanzania data set. And then, like I mentioned, we showed that the HBS1L MIP is actually, the drivers are actually on the HMIP2A and not both on HMIP2A and 2B. The polygenic score, which we constructed, shows that 5 SNPs from BCL11A and HBS1L MIP accounts only for 24% of HB variability. And like I mentioned as well, we found that the heritability for fetal hemoglobin was just 44% based on this data set. I would like to thank Dr. Stefan Menzel and Professor John Strobulis at King's College London that led this work. They were the PI on this project, and I was the one doing the analysis, and we had strong support from our PIs from Nigeria and then collaborators that will use their data set for the meta-analysis. So we are grateful to Stewart Nkumbe and Dr. Siana Mitatiro from Tanzania, and other colleagues from King's College, Helen Cates, John Browin. From St. Jude Children's Research Hospital, we got data set from them, and we are grateful to our collaborators there. And also from Nigeria, Professor Titilope Adeyemo, Professor Obiageli Inodu, and Professor Hafsat Ahmad, who possibly might be on the call this afternoon. We're also grateful to our colleagues at University of Oxford that helps us with the functional GCTA that we did. And I'm grateful to my PI currently at Queen Mary University of London, Professor Segun Fatumo, and members of our team at Queen Mary University of London. Thank you very much for your attention. And again, thank you to Helios for inviting me to give this talk.
Thank you very much, Shola. That was absolutely fantastic. Really, really interesting. We now open the floor to, and you get lots of lots of applause here. I don't know if you can see it on the screen. So really well done. Thank you so much, Dr. Oyesola. I'm sure there'll be lots of questions, and you now open the floor to questions. If you could either raise your hand or put your question in the chat down to personal preference, we will then get you to share some questions. Okay. We have a question from Mohammed Nuruddin. If you would like to, let me just allow you to speak now. Can you?
Yeah, I can
ask your question, please. Thank you.
Yeah, thank you, Dr. Oyesola, for your presentation so far and your stride in the sickle cell sphere. I'm just wondering, your study and data covers 3 main sites in Nigeria. And you would agree with me that those 3 sites are not completely representative of the ethnic and biological diversity of the Nigerian population. Is there any plan towards expanding the population from whom, sickle cell population here, from whom your data has been taken, so that a wider view of the genetic composition of the potential regulators of fetal hemoglobin would be better captured and would make your deductions more robust than what we have at the moment? And if there are plans, are there opportunities for wider collaboration like you have had with Professor Adeyemo and co? Thank you.
Okay. Yeah, thank you, Mohammed, for your great question. Yes, I agree with you that the data set we have has not captured the vast diversity of people in Nigeria with sickle cell disease. So yes, we have plans to look to collaborate more widely than what we have done, so that we can capture as much as possible a lot of people with the disease across different ethnicity, and which could give us better picture in terms of fetal hemoglobin or some other clinical phenotypes of sickle cell disease. Yes, we have plans to do that. And yes, we are hoping to collaborate. For instance, the 3 clinicians we are collaborating with in Nigeria, we're still working together, and they are also part of the SPACO Nigeria, which is led by Professor Unodu. And then with SPACO Nigeria, they have different sites within the country and different clinicians that they work together. So we hope to leverage this for our subsequent studies. Thank you very much.
Wonderful, thank you, Raj. We have some more people asking questions. Olawale, would you like to ask your question next, please? And I activate the microphone. Olawale, could you please ask your question? Unless you prefer to put it in the chat. Hello? Maybe we go to the next question first, just in case there's some technical issues. So we also have a question from Mustafa, please. If you would like to ask a question next, we then activate your microphone. Hello? Mustafa,
Yeah, can you hear me now?
yes, that's perfect.
Yes.
Wonderful. Thank you.
Dr. Ojewunmi, nice presentation. My name is Mustafa Abasi. I'm currently working on sickle cell disease as a PhD student. That was an excellent paper. I read your paper, went through it. Was really wonderful. Just a quick question. I have a couple of questions, but I will just start with one. That is the missing heritability. I just want to know, how does your data contribute to this missing heritability? Because what I've learned is the heritability there was based on those two genomic region. They were able to explain 24% of the fetal hemoglobin variation in the population that you studied. So how does that data, comprehensively looking at this data set, how does it, what can we learn, or how what has it added to our knowledge in terms of trying to understand the missing heritability of fetal hemoglobin?
Yeah, thank you very much for your question. You could see that in my presentation, when I was showing the rationale, I said that we have less than 30% of genome-wide significant heritability for fetal hemoglobin. And from our study, we just got 24%, which is similar to what's been published previously. So in terms of heritability, it didn't contribute much because it was also showing the picture that we have seen before from previous studies. But what that shows clearly is that one, like I answered the first question, we might need to increase the wider spread of the data of the patients. If we're going to look at fetal hemoglobin, if we make it more diverse, maybe we could capture new variants. But much more is the fact that there may be some rare variants, okay, that we are not able to detect because they have smaller effects, okay? So when we do rare variants analysis, we possibly might be able to find them.
Okay, that's excellent. So in your data, you also saw additional 3 loci that were found to have some association with the fetal hemoglobin. What can we learn from those three genomic regions, like SLC?
Yeah, SLC on chromosome 9, TICRR on chromosome 15, and PIZO2 on chromosome 18. Unfortunately, when we looked at this data set in the replication data set, which was data set we got from the US, Tanzania, and then the UK SCD data set, those first two SNPs on chromosome 9 and 15, they were not replicated. Okay, so when we looked at chromosome 18, the one for PIZO2, we saw that replicated. Okay, but at the moment, we really don't know what they do. We only saw that it is replicated, but to take it further, we may have to look at some biological experiment to see whatever this PIZO2 might be doing. But at the moment, we really don't know what it does.
Yes. Thank you. This one is just a comment that the malaria gen data set has some sickle cell, has some information about Gambian population with some sickle cell disease, and even the cardiac population. Next time we might look into that data set to kind of enrich your diversity in the study population.
Which data set is that again?
Malaria gen.
Okay.
From Gambian, which I'm currently looking at to try to understand the genetic background of the sickle cell patients in the Gambian population.
Okay.
So this is just a comment on that.
Okay, thank you.
All right, okay.
So we also have another question from Oluwale. Maybe we try again to see if the microphone works this time.
Good afternoon, Dr. Oyesola. Can you hear me? Yeah.
Yes.
Thank you so much. That was a beautiful presentation. I'm currently a PhD student at the University of Ibadan, and I'm developing a deep learning model for classifying sickle cell anemia severity. And I think if I'm correct, what I learned is that the better the level of the fetal hemoglobin, it means that the survival rate of the patient, the patient has a better survival rate. Is that what you're trying to say? Oh, fantastic. I can't hear you. I think you are muted.
Oh, okay. Yeah, I said yes, that's what we would expect. That if fetal hemoglobin is higher, it should improve survival, it should increase the quality of life. Yes, that's what we would expect.
Okay. Fantastic. Because I actually had a conference which I did at Lagos University, and we actually did an experiment talking about classification of hemoglobin level in sickle cell anemia patients. So I think you have really actually verified that what we did we're actually on the right path. Then my second question quickly, so I don't take your time, is how available is your data set for now? I know it's just a new study. Will you be providing data? Will your data set be made available? Because right now I know the constraint of actually getting data, especially local data in Nigeria. That is what I'm facing now at methodology level, and it's a bit difficult to get local data sets.
Yeah, so this data set we generated, we have in terms of the ownership, we have the great contributions from the clinicians from Nigeria. They have access to this data set. We generated this data set together. But beyond this group, the data set is not available in the public domain because of the ethical concerns that we got while doing this study. But if anybody is interested in this data set, they can write to me or to any of the collaborators from Nigeria or to Dr. Stefan Menzel. We'll look at the requests, and then, yeah, it's possible to share the data if we really know what they want to do and what the request is all about.
All right, thank you, Dr. Oyesola. Thank you so much. I appreciate it.
You're welcome. Yeah.
Thank you again. So we got a few more questions in the chat. I just read them out now. So thank you, Sola, for this fantastic talk. I wanted to ask you, did you check for correlation between the SNPs and clinical complications?
No, I didn't check for that. I only focused on fetal hemoglobin. But of course, the same data set could be used to check for other clinical complications.
Thank you. We also got another question from Sotirula, who says, amazing work. Thank you for the great presentation. Does the analysis include SNPs failing into non-coding regions that could be perhaps explored through epigenetic studies?
Yes. So the regions we have for the BCL11A and for the BS1L MILK, they are intronic regions. And yes, there are different data sets which could be used to further interrogate them, including epigenetic data sets. Yes, I agree. But we didn't include that in the study we did.
Okay, wonderful, thank you. And then also a question for me, because you mentioned that the SNPs identified explain 24.1% of the HbF variants. And what are the challenges to identify the variants explaining the remaining HbF variants?
Yeah, thank you for your question, which is also similar to what I said to one of the persons who asked question earlier, is the fact that this is the second GWAS conducted in Africa. The first one in 2015 from Tanzania cohort, this one from Nigeria, which was published earlier this year. I believe that if we increase the number in terms of the sample size, we possibly might find a new loci. That's one. The other one is to look at rare variants, which may have large effects, okay, which can explain why we still have this missing heritability, because like half of it is still missing. So if you look at that, doing rare variant analysis, we possibly might be able to find some genetic variant that are associated with fetal hemoglobin.
Wonderful. And can you explain why the percentage is higher in the non-anemic population?
Yes. So, for instance, the non-anemic population, which were the twins data sets, they got about 45% from genome-wide significance. And then from our own study, similar to what other studies have reported, about 24–25%. One is that the allele frequency are different. So when the allele frequency are different, then the heritability also might be different. So that's one of the things that you might want to look at. For instance, for the HBS1L MIP on chromosome 6, we have quite a number of SNPs within that region that have higher allele frequency in the European population, whereas, as high as about 20%, but in the African population, you will see about 3 to 5%. Okay, so this also is one of the things that could affect heritability.
Wonderful, thank you so much. I'm not sure if there are any more questions, but questions might also come up later. So what is the best way people can reach you? We actually have another question from Oluwole Alivide, and I apologize the pronunciation. Are you still unmuted from earlier? Oluwole, can you ask the question again?
Sorry, doctor, can you hear me, sir?
Yes, I can hear you.
Yeah, I just wanted to say, if it's possible, if you could share your contact, so that if we want to reach out to you, I don't know if you don't mind. If you could share that on the chat box.
Okay, let me try it
much.
if I could. I just put it in the chat box. Did you see it?
Yes, sir. Thank you very much.
Yeah. Okay.
Wonderful. Thank you so much, Sola. So if anyone else has any more questions at a later stage, and maybe if you haven't had a chance to read the publication yet, I highly recommend you do so because it's very exciting work. Sola obviously currently shared his contact details in the chat. So if you have any more questions later, I'm sure it would be fine for you to contact him at a later stage. Sola, thank you so much again for this really wonderful presentation. It was a real pleasure, particularly since it's the first speaker series. So thank you so much for making it such a lovely start. If you haven't had a chance to watch this webinar today, or if you want to rewatch it again at a later stage, this webinar will be recorded, so you can watch it anytime. And feel free to share it with anyone who you think might be interested in this. It's been absolutely fantastic. So thank you so much, Sola. Again, if you have an interest in other webinar series coming up in the future, please go to the heliosaction.eu website, to the webinar section, and then you can register for each individual webinar on the website. So thank you so much. Well, we look forward to continuing the discussion, and I wish everyone a lovely rest of the day. Thank you.
Yeah. Thank you for having me.
Thank you.
Bye-bye.
Bye-bye.